Okay, sorry, I was like, okay.
Yeah. Hello, everybody. Hello, professors, dear friends, all. I'm Dr. Amal Amin, Associate Professor at National Research Center Egypt, founder and chair of Women in Science Without Border Initiative. It's my pleasure to have you all here in our first webinar of the series of Saturday Science webinars, with the theme of challenges facing humanity at the age of COVID-19 and beyond. It will be a regular activity for Women in Science Without Borders, which was founded in 2017 as global template distributed in 48 countries with the slogan of Science for Sustainable Development. The aims of these Women in Science Without Borders are to develop society with science, to make science available for all, and to connect people and the world with science, and to include scientists, policymakers, industry, NGOs, and so on in one template, and to empower inclusion. We are not for women, we are for all. So that we have here mixture always between women and men, because we are all scientists, we have to cooperate with each other. The participants at these webinars, the first one, are mixture from males and females in different percentages coming from 61 countries. In addition to Egypt, our partners in Saturday Science webinars will be SciComm X, which is Science Communication Agency, with a special thanks to Dr. Saad Lotfi, its founder, and his colleagues. And the second partner is Science and Development Network MENA Region Edition, SciDev, with special thanks to its head, Mrs. Busaina Osama, and co-workers. Today it's my pleasure and honor to host 4 great speakers and friends as well. And all of them, fortunately, were participants of previous WISWAP meetings. The first speaker will be Professor Dr. Quraisha Abdul Karim from South Africa. The second speaker will be Professor Rami Karam Aziz from Egypt. The third speaker will be Professor Dr. Maria Augusta from UK. And the fourth speaker will be Dr. Mitsunobu Kano from Japan, who will be our speaker and moderator, both for the discussions at the end. First, I will give each speaker the chance to give a short talk for 15 to 20 minutes. After my short personal introduction for each of them, then we will start answering questions. The questions may be dropped in the chat or Q&I, both of them. At first, it's my pleasure to introduce first speaker, Professor Dr. Quraisha Abdul Karim from South Africa. She is a professor in clinical epidemiology, Columbia University, USA, and infectious diseases epidemiologist, and associate director of CAPRESIA, South Africa. She is member of the African Academy of Sciences, the Academy of Science of South Africa, and the World Academy of Sciences, TWAS. Also, she is L'Oreal recipient. Her lecture title will be The Dual Epidemics of HIV and COVID-19. You may start, Professor Quraisha. You're welcome. Sorry, the sound is mute.
Okay. Thank you very much, Professor Mal, for creating this opportunity for all of us to gather on this Saturday afternoon under very difficult and challenging conditions of COVID-19. And I also want to thank you for that warm introduction. I want to acknowledge and recognize my co-speakers in this session and other distinguished guests. I'm also pleased to see in terms of participants, there are lots of participants from all over the world, and really looking forward to this series of webinars, I think. I'm just checking if I have share screen, because I'm talking while I'm hoping my slides will come up. Is that can you see my slides?
Yes, I see.
Okay, excellent. I have to say that I'm not the most technologically competent person, so this is going to be fun, and I hope in the next 15 minutes or so to really talk to you about the dual epidemics of HIV and COVID-19 and opportunities they present for synergistic approaches. And I think one of the reasons I chose to focus on HIV and COVID-19, and while I will draw mainly on my experiences living and working in South Africa as a scientist, that these are epidemics that they're not just epidemics in South Africa, but they really challenges for sub-Saharan Africa, and they actually are global challenges. By way of overview, I'm going to quickly talk about the HIV-TB epidemics, how the government responded to COVID-19, and some of the infrastructure in our HIV response that was tapped into to enhance our response in a very rapid way. And then to close with reflecting on some of the challenges and opportunities that we have as we continue to grapple with these dual epidemics globally. So if we look at HIV globally, we have about just under 1 million deaths that continue to occur, despite access to triple therapy that has changed AIDS from a inevitably fatal condition to one that's chronic and manageable. We still continue to see 1.7 million infections taking place just in the past year globally. And we have just about 38 million people living with HIV. There are still many challenges that continue to face us in responding to the HIV epidemic. Just looking at the figures that I shared, now I'm going to move a little bit into the situation in South Africa. In many parts of the developing world, we see an intertwining of the HIV and TB epidemics. So I'm talking about them together because it's one of the most common opportunistic infections that we see with advancing HIV disease is TB. So if I focus now on South Africa, it is among the worst affected countries for both HIV and TB. And although we are home to less than 1% of the global population, about 1 in 5 infections that occur globally is occurring in South Africa, and that translates to about 7.7 million infections. We also have the 4th highest rate of HIV-TB co-infections, and about 60% of the new TB cases that we see is from advancing HIV disease. What we have seen, though, is government responses, particularly from about 2007, has resulted in about a 1/3 reduction in AIDS-related mortality, and TB death rates have been halved from 224 per 100,000 population in 2010 to 110 per 100,000 in 2018. And a lot of the success can be attributed to political commitment, collaborations and partnerships across the world, investments in training and capacity building for health service delivery, but also for research, building and strengthening our ability to undertake and lead research, building diagnostic capacity, surveillance capacity, and very importantly is how community engagement and partnerships has brought synergy between science and activism. And there's also been some good examples of innovation for drug delivery and adherence support. And I could spend a lot more time, but that's just to give you a bit of a flavor in terms of the magnitude of the epidemics and also what are some highlights in our responses. Moving on to COVID-19. So South Africa, very similar to Egypt and to Nigeria, we're one of the first countries in Africa to see cases of COVID-19, and these initial cases were imported from international travelers, And then we saw a spread initially with contacts of those travelers and some nosocomial transmission amongst healthcare workers. But overall, what happened was South Africa responded very quickly, very early on. We had very, restrictions on public gatherings, schools closures, and banning of international travel. And there's a whole lot of other things that were done, including the institution of a national lockdown, an extreme form of social distancing around the end of March. And you can see that where South Africa was by around early May, it was clustering with those countries that were responding and controlling and containing the COVID-19 spread. And we all know that we can't stop the spread of the virus because none of us have immunity, but at best what we can do is slow down the spread of the virus so that we can better prepare health facilities in terms of those cases that develop disease. And you want to make sure when those numbers increase, your health facilities are not so overburdened that you can't provide good quality care to those coming into hospitals, and making sure if you have good quality care, you can see that mortality rates would be lower. So if we move on then to just a few weeks ago to August 2020, we see South Africa is now right on the top. And part of this is the reflection of some of the planning and preparatory work being completed. And as we've moved down to more easing of lockdowns, the kind of anticipated spread of the virus started to occur. And while we are up there in terms of the UK and the US in terms of numbers of infection, what's important is our mortality rate has remained fairly low. Now, what are some of the things where and why our response was so successful and contributed to this better preparation was the investments that were made in HIV and TB. And I'll just dwell on a few of these. And the first is diagnostic tests are critically important. And it is quite amazing when we think about the COVID-19 epidemic from the first reported cases late last year, that already within a month we had the full genome sequence available. Shortly thereafter, we had diagnostic tests available, and our ability to test has been a very important part of the response. And in South Africa, we had a very strong a viral load, a PCR platform for HIV viral load testing that we were able to quickly use and utilize and expand to do SARS-CoV-2 testing. And so it's one thing having the equipment and the trained personnel, and then it's the other, which is in epidemics, you want to do wide-scale testing. And here you see the National Health Laboratory Service that does the national testing, the capacity being quite limited very early on in the epidemic, and how that grew. And as that capacity grew, we were able to do more than what most countries did, which is mainly test people who are coming in sick at health facilities to do active case finding. And the active case finding that was undertaken enabled us to not just wait for cases to arrive in the hospital, but to find the cases in the community and then find the contacts of those cases, refer these individuals who were infected to hospitals earlier, those who were asymptomatic, if they couldn't self-isolate, put them into government-provided facilities, etc. And that's a very typical public health response to epidemics. And you can see while early on the positivity rates of the tests were low, as we reach surge and the peak, that started to increase. But there was also another challenge that we faced is once we built the capacity and started to do active case finding, we also started to compete globally for the limited number of test kits that were available. We're also competing for reagents, etc. And so the testing strategy needed to be adapted. And because of the shortages in test kits, we had to focus on those mainly being admitted to hospital. And I think that's one of the reasons why you're seeing this higher test positivity rate. And then as we now in the decline of the epidemic and fewer hospital admissions, etc., we're starting to see that coming down. So the second part was around, as I mentioned just now, the importance of finding the cases and not waiting for cases to come to the hospital. And, you know, that's what we saw a lot in the UK and the US to some extent in Italy was when you have just wait for people to arrive when they're sick, then you miss the opportunity to actually respond earlier. The death rates are higher. And one of the things we have is a huge cadre of community health workers that have been part of our HIV and TB response. And we're able to build on this partnership of involving local community-based organizations and leadership to do home visits. And within literally a few days, we're able to mobilize something like 80,000 community health workers to visit over 11 million homes, particularly those that were most vulnerable. And they were able to do a no-touch screening and asked through asking a set of questions. And then if they were symptomatic, trigger referrals for testing. And if they were sick, initiate quick referrals. And that enabled us to find a lot of cases very quickly, do some of those. What I show here is the epidemic curve and how the responses that we put in enable the slowing of the spread of the virus, but not stopping the spread of the virus, which we can't because of the lack of immunity or vaccines, and also show you hospital admissions, etc. In summary, the early lockdown in South Africa enabled us space and time to improve diagnostic testing capacity, active case finding, and strengthen the COVID-19 healthcare capacity. And this is possible through strong political commitment and leadership from government, strong public-private partnerships, and also building on the existing HIV/TB infrastructure and resources that were available. And that's important, I think, because we in Africa and in many developing countries are dealing with multiple epidemics simultaneously. So while doing so and enhancing our response to the epidemic, we found the essential health services like HIV, TB, and non-communicable diseases started to be compromised. And this was partly because patients were scared to utilize services in case they acquire infection, but also as the number of COVID cases increased, there were restrictions in who could utilize health facilities. Another casualty in this prioritization of COVID-19 was the stoppage of all research that was underway. Important other point is, as usual, we find gender comes up strongly, access to sexual reproductive health services, including supervised delivery of babies and pregnant women. We find those issues were compromised, not viewed as essential services. And also the kind of implications of lockdown was more than around just the restriction of movement, but also on jobs, and the socio-economic impacts were disproportionately higher for women. We have more women-headed households. Women tend to be working as first responders rather, but also tend to be self-employed. And we had a challenge the economy that was exacerbated and exaggerated in terms of its impact on women. And then while we've seen quite unprecedented scientific progress and global collaboration and partnerships in terms of vaccine development, diagnostics and therapeutics, I think what was important in Africa is that we joined the queue, the global queue for limited resources, and these restrictions by not playing a leadership role had a huge impact. So I think as we move forward and as we all learn to live with the virus, there are some important considerations in the opportunities that we have. And I think while many of us have the privilege of having connectivity to join a meeting and a webinar like this, there are more people that don't. So these epidemics occur within, and pandemics, within huge disparities within and between countries, and where the most vulnerable reflect an intersection of multiple factors, not only disease burden, poverty, access to services. While we see resilience, I think the opportunities are not being tapped into adequately for sustainability in terms of community partnerships and moving from anxiety to agency. And I think that's an important lesson we learned in HIV that's not being translated in COVID-19, is how do we enable and empower communities through knowledge and through resource allocation to really have more agency in responding to just another threat in our lives. There's also, by our very vertical opportunities, we are missing the opportunity to link HIV, TB, COVID-19, and non-communicable diseases in a more synergistic approach. And again, there's a lot of preventive, promotive health activities that can be undertaken if we integrate our approaches by approaching it as a single condition and separating COVID-19 from everything else. We're missing opportunities where these comorbidities impact the disease trajectory of COVID-19, but also individually important and understanding what those interactions are. And I think if we use this more synergistic approach, and look not just at disease presentation and health service utilization, but working from the ground up and from bottom up, we have good opportunities to look at preventive promotive health services and early diagnosis and linkages to services with better outcomes in terms of saving lives, but also ensuring lives that are being lived are healthy. And I think it's also another reminder for us in developing countries and for our governments to invest more in science, technology, and innovation. We've paid the price in terms of entering and being part of the global competition for diagnostic tests, including personal protective equipment, reagents for PCR, medication. And as we know, if we don't step up, because we're not leading vaccine development, when vaccines become available, we'll also be waiting for the crumbs. So I think some of these lessons, this is the 7th coronavirus that's affected humans. The greatest severity has been with SARS and with MERS, but restricted impact geographically. But there's no question that as we move forward, we will be faced with many more epidemics and many more challenges. And how do we use this time and opportunity to better prepare for future epidemics? And that as we live with the virus and respect and utilize the non-pharmaceutical interventions we have of hand washing, barriers like masks and social distancing in different forms. How do we balance living with the virus, saving lives, saving livelihoods, but also importantly, which I didn't touch on, was how some of these mandatory interventions on social distancing have also increased violations of rights of individuals. And I'm not talking about tobacco sales and alcohol sales, but really the enforcement of some of the lockdowns and how people were treated. So we've got to think about rights, we have to think about livelihoods and lives. So thank you very much for your attention, and I'm happy to take any questions.
Thanks a lot, Professor Quraisha, for this nice talk. Really, it's very nice talk because it's a nice advice from wise scientists that living with virus. 1 minute. It's very nice advice from wise scientists that living with virus, balancing, saving lives, livelihoods, and protecting rights. It's nice advice. So thanks a lot for your lecture, and we will transfer to the second speaker. The second speaker is Professor Rami Karam Aziz. Professor Rami is currently professor and chair of the Department of Microbiology and Immunology at the Faculty of Pharmacy, Cairo University. Professor Rami is recipient of awards from Tuwaz Aru, and the Egyptian Incentive State Awards. Professor Rami's lecture title is Preparing for the Next Pandemic, the Better Lessons Learned from COVID-19. You may start, Professor Rami, for 20 minutes, from 15 to 20 minutes. Thanks a lot.
For this, yeah, I will speak and just 1 minute before, let me know. So I will, I will not take so much introduction. So first of all, thank you so much for this invitation, and for I'm very happy to be with this excellent group of international scientists, and thanks for everybody who are spending their weekend with us. I will share now my screen, and it's really nice that the last talk was just ending with how to prepare for the next pandemic, which is actually the title I put for my talk. And this was, so can you see the screen now?
Yes, we can see this.
Okay, great. So I think this title, I put it a while ago, not knowing if we will be able to even survive this pandemic or not. But of course, we have to always stay on the hopeful side. So preparing for the next pandemic. And my talk is divided majorly in two parts. The first part may be a little bit more theoretical, but more general on how to be prepared for any possible pandemic. And then in the remaining last 7 or 8 minutes, I will talk about some examples from my research, but I understand this is not like a data heavy session. So I'll just give you a few examples on how we deal with one of these expected pandemics. So first of all, let me assume, I mean, I'll just make some assumptions, and that everybody listening to me here is human. Maybe some of us have their pets with us, but I assume there are no robots or anything. So if you are still listening to me now at this moment, if you can hear me, you are one of those lucky survivors. First, I would like to thank you for listening to me, but actually what I want to say is that if you have made it so far in humanity, what we say in Arabic is Mabruk, congratulations. This is not for granted. Just 200 years ago, the mortality rate among humans was much higher than what it is now. Well, the ultimate mortality rate is 100%, of course, but in terms of life expectancy, so now the average life expectancy around the world, maybe it's highest in Japan and it's lowest in some some sub-Saharan African countries and in Afghanistan, but on average, humans would easily live more than 65 to 70 years. This is not granted children survival. Like now people just go and give birth, and this wasn't the case a long time ago. So now we are blessed and happy. So I want to say, if you are here so far, you have to be congratulated, and you have to be thankful also to the higher power you believe in, or to your genes, your ancestors' genes that were moved to you, because we are all survivors here as humans. Actually, every biological entity on the planet is a survivor of some sort. A successful genome that managed to survive among all the chemistry around us, but also, especially as humans, we survived a lot of infectious diseases. Our genes are not randomly distributed, what I want to say, among the population. Some populations have been wiped out by some diseases. And we have also to be very grateful to our diversity, genetic and just in general diversity. If we were clonal, one virus would have wiped us all, a long time ago. So this was just what I want to tell you. Whatever your specialty is, this is a very easy concept in biology. If you're here, you have been naturally selected to maintain the species, whether you want it or not. You are carrying a great mission of surviving and reproducing, not just for your sake, but for the sake of the species. And even if some people are depressed and they don't really want to live anymore, but this is what all our genetic program for millions of years have been designed, have been evolving, just for this incredible instinct of survival and passing our genes on. All right, let me move to our topic now. So a very interesting book called 12 Diseases. I used to see it every year when we used to go to conferences in the American Society for Microbiology conference. It's called 12 Diseases That Changed Our World. And 10 out of these 12 diseases are infectious diseases, and 9 of them are human infectious diseases. So the book is, of course, it's in the form of history or storytelling, but it's also a very interesting book about how our genes and our population have been shaped based on epidemics. For example, the plague that infected Europe and most of the world at that time, the old world before the discovery of the Americas, to those who live in Europe, there was no Western world. So this plague wiped out a lot of people. And when you look at our genes now, we have genes called the HLA genes, the human leukocyte antigens. These genes are not just randomly distributed. Some, I don't want to get too much into details, but some genetic variants have disappeared because they were the most susceptible to this disease. If we were all twins, if we were all, identical genetically, we could have been, as I told you, we could have disappeared. But because of our very high diversity, and the highest diversity in our system is our human body, is the immune cells. That's why just going back to the past speaker, that's why we see so much discrepancy in human response to this coronavirus disease, because we just don't know how the immune system will respond, even if you have all equal opportunities in therapy and everything. So these diseases, again, plague, tuberculosis, syphilis, malaria. Malaria is still shaping the world. Tuberculosis and malaria are the biggest killers of humans among all the infectious diseases. Cumulatively, AIDS is a new epidemic, of course, just like less than 50 years old. Cholera, there is a lot of waves of cholera and so on. And now they are affecting our present. Again, I don't need to say anything. As they say, no caption needed. You just know, just look in the mirror, just look at our daily lives. I don't think we are getting used to it yet, but it's much better than 3-4 months ago. Everything happening now is beyond belief, right? We cannot just travel easily. Some people cannot even be allowed to travel if they want to, because they don't have any urgent reason to travel. Schools, my son is now attending his class online, probably affecting my internet as well. And we don't know when life will be back to whatever we used to call normal, which is our normal, not the normal of our ancestors anyway. And the future, which is the topic of the rest of my talk, the future is also shaped by these infectious diseases. So it is expected that what we call the AMR, antimicrobial resistant pathogens, may be killing more people than cancer. Cancer is killing 8 million on average. Infectious diseases are likely to come back with the resistance to antibiotics unless we get prepared, and that's the topic. So we will see all these titles in the news. These titles were all over the news before COVID-19, but believe it or not, even COVID-19 as a coronavirus, there were, as many of you have read, warnings that this is going to happen. So we have to learn the lesson, as in my title. The bitter lesson is that, as we say in Arabic, لا يلدغ المؤمن من جحر مرتين, and in English, what is said is like, fool me once, shame on you, fool me twice, shame on me. So we have been fooled a dozen times, but we shouldn't be fooled anymore, right? I just want to get a very quick flashback. In November, it's a very interesting coincidence, or maybe we had a conference, Arab American Frontiers Conference here in Cairo. It's in a different Arab country every year. And I proposed a session with my co-host from the US. It was an infectious disease session. And there was kind of criticism, why you want to talk about infectious diseases in 2020, right? It's almost gone. We said, no, it's actually the coming threat. And we were just 2 months short of the real prediction. And interestingly enough, this talk by Ely Graydon, the lady that you see, whose picture you see on the left, and other faculty member, it was very interesting because she spoke about pandemics, how to prepare for them, about animal spillover, and other colleagues spoke about wars, how wars and migration. Like I'm going to summarize this a little bit now. So how to prepare for the next pandemic. First, know where they are coming from. One of the major, and we learned this, I'm sure, very well now, sources is the animal spillover. So animal spillover is what you see from bats, from rodents, from chicken, from birds in general for the flu. And the other one is human migration. And there are so many reasons for forced migration: wars, which we, water shortage, famine, armed conflicts, another name for wars, and all like not just wars, but like local problems, civil wars and things like that. And the third source is antimicrobial resistance, which is more related to my work. But it's very interesting because we have to be prepared for all these. We have to keep monitoring them. So how to be prepared? It's the topic, right? So one thing is a concept that has been here for a while. It's called the One Health concept. We are humans living with animals in an environment. So these three elements, 1, 2, 3, human, environment, and animal, you cannot separate them. You cannot say I'm a zoologist. You cannot say this is medical school and this is veterinary school, and we don't care about climate change. No, everything is connected, as we have seen just exactly in the COVID-19 case. So these viruses came from animals, bats, and why are we getting bat diseases? Because of environmental changes, because we are getting into bat territories, we are cutting their trees, we are just building where the bats live, we are eating things we shouldn't eat, and so on. So what we need is global alliances. No country will do it alone, not just because of economy. Even if you're the richest country, you have no reach to, for example, if in the US, you need South Africa, you need South Australia, you need South Brazil to see what is, and of course China, you need to know what is emerging in every part of the world. Genomic, metagenomic preparedness, I'm going to talk about in the rest of the few minutes remaining. What I want to add here, specifically what we learned from SARS-CoV-2, from the coronavirus, we have to be diagnostically prepared. It's so embarrassing that we were not as well prepared. We know how to, I'm always saying we as scientific community, we know how to prepare antigen tests, we know how to prepare antibody, but we were not that ready. We knew there is a coronavirus coming eventually. We were not that ready. So I'm thinking that everybody now should be ready with the test. Just what is needed is the virus or the bacterium to put on the test. Just have the paper strips ready, have the PCR ready, have the reagents ready. It's worth spending, even if you spend a couple of $1 billion. That's nothing. That's what we're losing I don't want to say every day, every hour now because of the pandemic. So just prepare tests and just, wait to know what is the pandemic, so this is what is done every year for the flu now, influenza. Every year there is a vaccine prepared for the next year before knowing what is the next strain coming from influenza. Once this strain is detected in Hong Kong or in Australia, they send it around the world, and all labs in the world work together to prepare it. So the flu is very well coordinated. We need to prepare for every other possible epidemic. We have to have our vaccines ready like now. Again, we cannot predict who is hitting us next, but we can, if we have all the production ready and just waiting for the signal what is the genome, because for SARS-CoV-2, it took us 3 or 4 weeks to figure out everything about its genes, what is the antigen. But we were, like, as a humanity, we were not prepared to produce that many vaccines to test them and so on. And what I want to say in the end, it's global leadership and coordination that is needed. And you have seen, it's really disappointing to all of us to see that every country until now has a different policy. The WHO doesn't have any, way to enforce. They just suggest things. They're not always right, but they're at least specialized. But different countries are doing it differently, even the richest countries in the world. And there is a lot of ego, unfortunately, competition. When there is competition is a crime now, It's not ethical. You just have to compete about helping each other. It's not ethical that one country says I'm producing the vaccine before the other. That's ridiculous. Just work together. There is no way here to compete. So I will just show you examples. They already exist. There is a global virome project, and its motto is preparing for the next. I didn't know that these slides existed before putting my title. I just went to their website. They're looking for all animals, sequencing their genomes and the viruses they have, building capacities and screening 500,000 of viruses. But believe it or not, their funding is being cut because some conspiracy theories say these people are building actually viruses instead of discovering them. If you go to a website for the PREDICT program, it's amazing. You just click on any country. Here, this is bat. In Egypt, they're sampling this bat from October 2016. So we know what is the risk in Egypt. Pigs from Vietnam, bats having coronavirus from 2017. Everything has been monitored by scientists, but nobody was listening, or maybe scientists were not shouting loud enough. And the last program I want to give again as an example is called the EcoHealth Alliance. Again, this program, believe it or not, their funding was cut by the US government, and they're now collecting donations. And guess what? All we need is like $50 billion. That's a lot of money, but it's nothing compared to what we are spending 8 trillion every day on public health to save lives. And 2 trillion every day on military expenditure. That's what humans are spending. So if you spend 50 billions in 1 year to sequence every possible virus from bats, you will not have the next corona epidemic. So with this, I end part 1, and part 2 is much, much shorter. I just want to show you a couple of examples from our, can you still hear me now?
Yes, I hear you. Yeah, just continue.
Sure, I will just share different slides. So now what kind of, like, all this is good
just
talk.
Share screen, Dr. Ram.
Yeah, sure, I will. So I have about 5 minutes, right?
Yes. Just
okay,
share
great.
the screen.
Sure, I am. So all what I said so far is very good, but my job is a microbiologist, and I am a microbial genomics expert. So I'm not a policymaker. So I just talked to you about my opinion about policies, but now what we do, like in the lab, can we really do this? So you can see again, this is what I talked about in the past introduction. Superbugs are one of the coming pandemics. So the coming pandemic could be a virus or a bacterium. We call it superbugs, which means like Superman, like a bug or a bacterium that is resistant to almost every antibiotic we use. So this is almost established. Models are predicting this to happen in 2050, unless we either decrease the problem of resistance or we invent new antibiotics. And so far we are just, again, as scientific community, it's very slow to make antibiotics. It's not economically feasible because there is so much spending. And now with the COVID-19, again, it's not the highest priority. So, and it will be the same error again. So one way I want to show you a few examples from my research is using genomics and metagenomics. And you heard from Dr. Quraisha about it. It's one of the important tools, because in the old days, we used to see bacteria and viruses with microscopes or electron microscopes. But it's really hard to know much just by seeing them. So now by DNA sequencing, you can detect bacteria, viruses, and parasites everywhere without seeing them even by the microscope. Just collect swabs or DNA and look at what they do. And then you use the computer. So on the top right here, you see this huge tree is every coronavirus genome that has been sequenced to date. And they're divided into a few different variants, very similar to each other. No significant difference, but it's amazing. This it's like a dream for any genome person, you know. In 2000, when I was doing my PhD, there were about 10 or 15 genomes in the databases, 1,000 genomes around 2005. And today in 2020, just for the SARS-CoV-2, one virus, we have 95,000 genomes as of today. And they are divided every day, monitored from all countries, coordinated. It's an amazing work. And then it's like looking at archaeology and looking at papyrus. We try to bring these, you know, pieces of puzzle together to tell the story. So I'll just tell you that, so as I told you, we need global alliances. So one of the alliances I work with was looking at bacteriophages, bacterial viruses in the environment. And we discovered this using this method with colleagues from Netherlands and San Diego in the US, a new virus that is in all our intestine. We called it crasphage at the time. And monitoring it around the world, it's not distributed equal, but it's amazing because this virus is even ancestral to humans. So it was found in gorillas as well. So this is one example about one virus that is in, we didn't know anything about it until 2014, but we know now using DNA sequencing. So how about the superbugs I talked about? What can we do with them? Again, I don't want to take so much time. I will just give you examples. So this is example. Okay. This is one example from one of the teams I worked with. And anything you see in red is antimicrobial resistant bacteria, and green is still not resistant. So you can see that the red is winning. So we are warning you now, and we have warned you a lot. So I'm telling you, don't come in 2040 as humanity and say nobody warned us that be a genome. So what we do with the computers, again, is we keep reconstructing this. I'll skip most of my slides. I just want to show you another example from Klebsiella, which is, we have it in Egypt, bacteria that is in hospitals. It's resistant to all, we have 4 bacteria that were resistant to all antibiotics. And when one of these were discovered in the US, it was a big deal. Now we have a lot of this in many countries in hospitals, so we have to be careful. So again, I don't want to talk very pessimistically, but one of the optimism that I have is that I always show this picture. These guys now they are getting older. They were last year students, but now most of them are researchers or pharmacists. They built a huge database for antimicrobial resistance genes. And we used this in the German-Egyptian network for genomics to screen for resistance genes. So we are getting there. I will just jump to my conclusion quickly. So also, yeah, one of the things we did is looking at the hospital microbiome, and we got funded for this project recently by the Egyptian Academy, thanks to them. So we have money now. We have 1.6 million pounds, which is, it's not too much, but it's not a little. So it's $100,000 just to screen hospitals for bacteria. And now maybe we will look also for viruses. We looked in a very small hospital for very few samples, but now we can look for more. There is also the German-Egyptian networks. As I told you, it's all about network building. Nobody can do this alone. We also work with the International Global Sewage Surveillance. So we are looking at sewage, Magari, the wastewater. It's full of treasures, wastewater, not just for recycling, but you can tell everything happening in a country or in a city by looking at wastewater. What viruses are there? What are people eating? What bacteria they are shedding? What is the next resistance coming from? And so on. And finally, of course, everybody now is working on coronavirus. It's a duty to every scientist. So we're also trying to analyze the genomes to try to make sure no mutants will come from the virus that are more dangerous. We hope that the virus will mutate to something more peaceful. And with this, I thank you so much for listening. And I always like to acknowledge all people who funded us, ESRT, the Cairo University, and also everybody who participated in my scientific career since 1999. And thank you so much, Dr. Amal, for all your efforts and for bearing these 25 minutes with me. Thank you all.
Thanks a lot, Professor Rami. We heard the important terms in your lecture, and you are right. We have One Health, and we should have global alliances to cope with pandemics. It's good advice. So that we will transfer to the third speaker.
Thank you so much.
Thanks a lot. The third speaker will be Professor Dr. Maria Augusta Arruda. Professor Maria is Research Development Manager at University of Nottingham, previous academic staff for 13 years, 8 of them at the School of Life Sciences in Nottingham. She chairs the University Black and Asian minority network and interested in science communication and science diplomacy. Also, she is L'Oreal recipient. By the way, she is originally Brazilian and she transferred to UK years ago. But she made some collaboration between Nottingham and Brazil, and then she became a staff in Nottingham. Her lecture's title is Science for the Many, Not the Few. You may start, Professor Maria.
Thank you very much, Amal. And I'm going to share my screen.
15 to 20 minutes, Maria, okay?
I'll be as fast as possible. You know, I can be fast, Amal, and you can tell me off if I talk too much.
You have up till 20 minutes.
Thank you very much.
Okay, thanks a lot.
So I can't start my video, but I think I'll be able to start
just share
my presentation. Yeah.
The screen.
So hopefully. So can you see my PowerPoint presentation?
Yes, yes, yeah. I can see it.
Okay.
But just make it bigger.
I will make it. Hopefully, it's going to be all right. Just a second, please. Can you see me well?
Yes, yes.
Okay. Can you see the whole presentation?
Yes.
Okay. Thank you very much.
Not the few.
Exactly. Thank you so much. And I would like to thank my dear friend, modern friend, sister Amal Amin. We are sisters in science diplomacy. And my talk today is trying to explore a little bit more how interconnected the different aspects of life are. Sometimes thinking about science and talking about science, we detach ourselves from the wider society, and I would advocate for us to rethink this concept. And I will start discussing with you that COVID is a magnifying lens on inequalities. Right in the middle of the COVID-19 pandemic, Johns Hopkins School of Public Health released this article about trusting science and COVID-19. So I think that all of us here will understand and will unfortunately agree that this pandemic comes in a moment that trusting in science is in an all-time low in many countries across the globe. And unfortunately, that's what we are fighting against. But we need to understand what is the responsibility of science and scientists in this scenario. So one of the things that happened during the COVID-19 crisis is understanding the impact of inequality and racism, for example, on COVID-19. So when we think about even the developed world and countries, affluent countries like US and UK, we see that those that are marginalized, those that are kept on the fringes, in the fringes of society, are the ones that are suffering the most the effects of the pandemic. Many of them are health care professionals, physicians, doctors, surgeons, nurses.
Maria, sorry,
yes?
But please move the screen. The screen is stable.
Say it again.
The screen is stable. Only the title. We see only the title.
You can see only the title.
Yes. Just move the screen. The first page only appeared.
I'm so sorry. I don't understand what's going on.
You need to advance your slide.
Yeah, advance the slides.
Can you see now?
We see only the beginning, the first slide.
Oh, well, that's very, it seems that my screen is paused. So I'm going to, hopefully it's going to be okay now. Let's try again.
Yeah. Okay.
So can you see that now?
Yeah, okay.
Can you see
Yes.
Yes.
racism and COVID?
Yes, now it's green.
Okay, okay, fantastic, thank you. So racism and COVID, I told you before that, you know, a big chunk of those affected with lethal effects by COVID were members of the BAME community, of the Black and Asian minority ethnic community in UK. The same thing happened in the United States. The same thing happened in Brazil. So we can see that it's much more the effect of a white supremacist model than regional or economical differences between these countries. When you think about women as well, we live in a world, and that's very well described in this book, The Invisible Women, a world that is biased towards men. So we can confidently say that what we use today, all that wonderful, marvelous advancements of science were designed by Caucasian, Mayan, European, or North American men. It means that everything that we've got was especially designed to 8.85% of the world population. And when you think about who are the people in science, only 28.8% of the scientists in the world are women. So maybe that's why one can think we don't have an effective treatment for malaria, for example, but we do have quite an effective one for erectile dysfunction. It's a world that was not designed for women, by women. And that's why I think it's so important initiatives like the one championed and created by our friend Amal Amin, and now those that joined forces for the creation of the Women in Science Without Borders initiative. We do need more women. We need more women of the developed world. We need more men and women working together for a better future. And COVID is just one of the many examples we've got of something that knows no borders, risks that knows no borders. There are many complex scenarios like climate change, the ocean full of plastic. We are taking fish out, putting plastic in, and the humanity is really struggling to find ways of mitigating those events. Maybe because we still insist in working silos, we still insist in working on a national level when it's not sustainable, because these are transnational global threats. However, scientists, they realize that, and here we've got some mega structures. We've got on the top Sirius, which is one of the largest, or actually the most potent synchrotron light source in the world right now, that is based in Brazil. We've got Sesame here on our right, that is a multi-country megastructure. And all of us know the Large Hadron Collider and the CERN Accelerator Complex that is actually across the board of different countries, 3 countries, and that it's an infrastructure that is meant to have a global impact, and it does. So all of these things are very good examples of science diplomacy in action. And science diplomacy is an ancient affair. So if you think of the Silk Road connecting the Far East Asia to Europe, and how much knowledge and experience alongside goods worth were transporting throughout this route. And then when we think about, very prominent scientists, and here put John Kepler, that lived in many different countries across Europe, we see that it's the nature of the scientist to exchange knowledge, to go around, to be this ambassador. And that's why I keep saying that every scientist is a diplomat. They also understand that they've got an important role in showing the wrong things happening across the globe. So when Charles Darwin, the young Charles Darwin, went around the world and landed in Brazil and saw the way the enslaved people from African descent were treated there, he became a big advocate for anti-slavery movements. So we can see that we are not only scientists, we are an important part of the society, and we can make the greater good when we look not only on the hard science that is so important, but also how it affects it's in the middle of the society, and we can make the modern science diplomacy has got many instruments. The World Academy of Science is one of them. The Academia Brasileira de Ciências, the Brazilian Academy of Sciences, the Organization for Women in Science for the Developing World, among others. And the science diplomacy can sound something a bit ethereal for many of us, but it's actually a quite recent term that was first published in June 2009, and it is constituted by a holy trinity for science diplomacy: diplomacy for science, science for diplomacy, and science in diplomacy. If we think about the eradication of poliomyelitis across the globe, it is a very good example of science diplomacy, of science really showing it's without borders. During the very, very, the most active period of the Cold War, we had a US, an American scientist, and a Russian scientist joining forces to eradicate poliomyelitis across the globe, creating the vaccine that all of us that we are here today were vaccinated with. A very good example of science for diplomacy is the legacy of Professor Abdul Salam, the Nobel Prize laureate, founder of the International Center for Theoretical Physics, and also founder of the World Academy of Science. He identified this strip of land between the communist bloc and Europe, that is Trieste, the city of Trieste in East Italy, that borders Slovenia, that used to be part of Yugoslavia. He identified this region and built trust with ministers and dignitaries in the West and in the communist bloc to foster, to make it viable, the exchange of knowledge between these scientists that were isolated, that were kept apart by these political barriers. Here's another example: when the reopening of the US Embassy in Havana was preceded by a very intense collaboration between American scientists and Cuban scientists. The diplomacy for science is that discrete work that governmental and diplomatic officials to creating frameworks for the advancement of science. So in Brazil, we had this amazing program called Science Without Borders that sent abroad 101,000 students during 4 years. That it really changed the face of the Brazilian science technology landscape. We've got many other initiatives of this kind across the globe, like the Horizon 2020, the Global Challenges Research Fund, and the Newton Fund. Science in diplomacy is when science provides evidence-based advice for government. Obviously, the governments, they need to be amenable to these kind of advice. And I'm very glad that we've got here among us one of these scientific advisors. The COVID-19 outbreak has shown to the world how important it is, with the World Health Organization really trying to work with local partners to bring a coherent framework for the control and treatment of this disease. Science diplomacy is inside each one of us as scientists, but there are ways of harnessing this knowledge. And I'm very glad to say that many of the friends that I've got today, like Amal, many of these friendships, they were fostered during one of these courses in science diplomacy, like the one that is taught in Trieste, but also there are nowadays a number of resources online to learn more about science diplomacy and lead to the development of this framework. I'm going to finish my talk now with COVID-19 and science diplomacy. When we think about the development of vaccines, we know, the world knows, everybody knows, the companies know, they know that it can't be developed in a single country, thinking of a single demographics for people that look very much alike. We need to have diversity in testing these vaccines, and we need also to immunize not only the country that where that company or that government is, but we need to make a real effort to immunize the whole world, particularly when it comes to a pandemic like COVID that knows no borders whatsoever. So here we've got two examples. Brazil, I don't know how aware you are, is going through a very, very rough patch when it comes to trust in science and support to scientific endeavors. But we've got fantastic scientists that happen to be, that became very skilled politicians in the native sense of the word. You know, they use diplomacy to make things happen. And I'm very happy to see that my birthplace has managed to engage with these important programs for the development of vaccine, despite the government really not being that keen to make it go forward. And that's a very interesting article about how science diplomacy can help us to contain COVID. So Marga Gual Soler, that was one of our lecturers in the science diplomacy course, and Tulula Oni, that was also involved with the science diplomacy course initiative, they published recently saying that science diplomacy means international cooperation with science at its core. So and that's the beauty of the science diplomat that is within each one of you in this audience today. You've got the ability to influence and change the landscape, as long as you understand the important role that you're playing. I finished my presentation, and as you can see, it's below 20 minutes now, with this photo that I think epitomizes, you know, the warmth and the commitment that goes with science diplomacy endeavors. Science diplomacy understands that never mind the guidelines, the political arrangements, the differences, it is done just like science by people, and people need to be at the very core of what we do. And with that, I finish my presentation, and thank you very much for your attention, and apologies for the hiccups in the very beginning. Thank you.
Thanks a lot, Professor Maria. Yes, we need science diplomacy in such hard time of COVID-19 to increase global cooperation. It's a message for decision makers. I hope they will listen. Okay, the next speaker will be our moderator as well, Professor Mitsunobu Kano. Professor Mitsunobu Kano is a science and technology co-advisor to Ministry of Foreign Affairs of Japan, so that he is the right person to listen your message, Maria. He is professor and vice executive director in Okayama University. Dr. Kano serves as chair of SDGs Initiatives Planning Committee. He is in boards of directors in bodies for scientific activities, including Honda Foundation and Japan Society for Drug Delivery System. Dr. Kano had several awards from Tokyo University Medical School, the Japan Cancer Association, the Japan Society for Drug Delivery System, the Ino Foundation for Science, and others. His lecture is scientific thinking and real world thoughts from medicine and SDGs. Here, the microphone for you, Professor Kano.
Thank you very much, Amal, for your kind introduction. And let me, first of all, share my slides. Is it now visible?
Yes.
Is it visible for you? Great.
15 to 20 minutes, okay?
Yes, that's the trouble for me. So let me start. So I wish to cover 3 parts today. One is about the general science, and then the next is about vision of that with my own outputs. And then the third part is about somewhat bridging the science and society. So, excuse me. Now it's not too much. So, what scientists do? This is an analogy. Well, we are somehow traveling from known world to unknown. That's the work of scientists. When you might finding out something unknown by your intuition, and then you needed to go out for a travel, and that's scientific activity. And if you can bring in that into the known world, then you can be a hero. So the reason why I come to ask questions, some experiences of myself. One is about clinical in the hospitals, and then I served for scientists and also educator, and I met a lot of discrepancies in between science and society. So first of all, I wish to revisit what is the scientific thinking, but because sometimes it's not so clear for scientists themselves what are scientific thinking, in my view. So it's an activity to overcome our troubles. So the key steps in the science is about, first of all, we needed to know the issues or problems. Once we get into some specialties, it's not so easy to get the wider issues in the society or wider problems in the society. So we need to get back them, in my view. And you needed to ask questions. which is not only restricted to your own specialty, but also it's possible for you to ask any more questions. And some of them, of course, can be only answered by scientific activity, but it's really nice for us to think about, first of all, hypotheses about your own questions. And if you can prove them by data gathering, then that's of course scientific. But before that, it's really important. I wish to reinforce the importance of asking questions by knowing issues and problems and thinking about hypotheses. The reason why is that we have two systems in our mind, according to Daniel Kahneman. One system is about intuition. It's fast, but sometimes not correct. And therefore we needed to correct them by using system 2, which is slow but logical. And this is the scientific activity in my view. Actually, we humans may need some simple stories to understand the situation. But to make a story, especially when you are a scientist, then we need to think about utilizing the correct evidences to support the story. And if you, of course, falsify the evidences to make up the story, which is understandable, then that's not good. So we needed to think about the stories which are correctly supported by the correct evidences. So in summary, the general way of thinking in science is to get new ideas with more accuracy, which can improve our society. And this may be very important in the era of the SDGs, so-called, and also of this pandemic era, even with the era of climate change. We Japanese have recently have a very big typhoon hitting us, which might be very dangerous. And this is said to be due to the climate change. So in my case, my own or our own question was due to the clinical setting for myself. It was a pancreatic cancer, which is very notorious for very hard to treat. If you were diagnosed as pancreatic cancer, especially with metastasis, then the half-life is said to be, in average, a half year only. So we wish to treat them with some drugs, but it's really hard to do. So my question was, why? This is not infectious disease, but cancer, but I think it's another disease which we need to confront in our future. And in the case of pancreatic cancer, normally cancers are stained by some agent which is not opaque to radio emission, and then they are stained very whitish normally. But in the case of pancreatic cancer, it's not stained. And normally it's said to be because of the dead cells inside. Of course, there are dead cells, but actually there are a lot more live cells. And therefore I asked why. The normal way of thinking about using drugs in our body, intaken drugs may come to the target cells via blood vessel. And there may be no obstacle in between blood vessels and target cells. But actually, if you see the microscopic view of the disease site, then you can realize that there are a lot of things in between the blood vessels and target cells, especially in pancreatic cancer. There are some different type of blood vessel in pancreatic cancer, and also there are some fibrotic tissue which can harden your tissue. The blood vessel side of the story was that the blood vessel seems to have more cells outside than the normal cancer. And this is the middle is the normal pattern for the cancer. where you have only thin layer of cells around the blood vessel. And in this case, you can have a lot of drugs coming out of the vessels and then remain inside the cancer. But once you have some cells more outside the vessel, then you do not have much drugs coming into the disease site. And this may be the reason why you cannot treat the cancer properly. So I tried to use a drug to make the barrier of the blood vessel weaker, and then using some other new technology, a drug with the new technology, which is nanotechnology, and then showing that only when you have the both, the new drug with some alteration of the blood vessel structure, and then you can have a proper effect of the drug into the disease. And I further wish to prove whether the fibrotic tissue in the cancer can inhibit the transportation of the drug into the cancer cells. And the microphotograph here is showing the existence of tumor cells, which is surrounded by reddish and also blue parts, which are representing the fibrotic tissue, which is hardening part of the disease site. And you can see that the percentage of that area is around 60 in the true human tissues. So we wish to mimic that situation with the animal model, and then showed that the existence of the hardening part, fibrotic tissue, can actually inhibit the efficacy of the drugs. Now, therefore, we wish to decipher more about the thickness of the fibrotic tissue by using the 3D culture of the cells. For that purpose, we tested for the thickness of the fibrotic tissue by using the true human specimen. And then we revealed that the thickness is, in average, regardless of the stage of the disease, about 30 micrometers. So we tried hard by using the nanotechnology to make the thickness of cultured cells of fibrotic tissue to show the thickness to the thickness which we clarified with the human tissue. By using this model, we tried to use some drug which can inhibit the proliferation, the increasing of the number and the thickness of the fibrotic tissue, and then shown that the transportation of the drug through this thickness has been better by using these drugs, or inhibition of ROCK signaling, which make the fibrotic tissue more. And then now we are still trying hard to make the model which can build outside the body, even outside the animal bodies, to mimic the situation in the pancreatic cancer by using that technique, 3D culture, not only having the cells which are in fibrotic tissue, but also having the tumor cells in it too. And the co-culture, which is culturing the cells, not only the fibrotic tissue, but also tumor cells together in one, have this kind of structure. Red color shows the fibrotic tissue, and the green color shows the tumor tissue. And the ratio between the tumor cells and fibrotic tissue can now mimic the human situation. So we wish to utilize this model to decipher what kind of drugs or some other methods can treat this pancreatic cancer. So this was some summary of what we are trying to do. Although this has been not implemented in the society yet, but we wish to have this implemented in the near future. So thinking about the importance of science and also in job in general, what's important would be whether we can answer the needs from the society. The pancreatic cancer part has been, of course, needed by the patients who are suffered from pancreatic cancer and also families having the loved people suffered from the disease. But in the case of the infectious disease this time, it's also needed. And of course, I wished to answer that in a scientific way, but actually what I myself can be done was only the part, not the experimental research that we can do, but the part I wish to tell something scientifically correct to the society. So let me go through that part as my last part. So here's the material for kids in my country, especially, to speak about the correctness, correct information, scientifically correct information about the infectious disease this time. Because kids in my country, kids are only sometimes told what to do, but they are not allowed to ask why. Or sometimes people just can tell what they needed to do, but I think this is a nice chance for us to arouse questions for general public. So we wish to make that into this material. So we start the material from a question: how can we fight the new coronavirus? Let's think together. So this is our message. And also we attach a Q&A session by adding QR code to show some questions and asked by scientifically correct information. So the start of the material was explaining what coronavirus, and with some kawaii, lovely in Japanese illustrations as Japanese custom. And we show the size of the virus and also why we call this as coronavirus. The next thing about what are the symptoms? Because knowing the symptoms, you can see whether you are getting infected or not so soon. So we wish to make some explanation on it too. And then how people can get infected. And because of providing this information, you may be able to think about how you can save yourself from getting infected and also save other people getting infected. And here are some information about how you can wash your hands and why you need to do so. And also we explained how the infection can be propagated through clusters of people and how we can prevent from get infected by clusters. The last of the material is on the potential discrimination of the people who maybe get infected. There are a lot of miserable news about the infected people are sometimes discriminated, and therefore we thought it was very important to think about why we needed to love people who has been infected. So we pose a question: what is the difference between physical distance and emotional distance? And this material has been broadcasted by national newspapers or TV and radio shows or SNS. And also they are widely used in schools. And therefore we estimated that more than 100,000 people in the country might have used this material, which is quite a successful, I think. So my last question to you is, what would you wish to do now? And that could be an adventure for yourself. That adventure can make you a hero. And hopefully it should be by scientific activities. So this is my last slide. It's about, again, the key messages. The one thing is cherishing your own questions as a woman, sometimes as an African, or some people from outside of the Western cultures, like us, or of course, some people from Western culture, or sometimes as a younger researcher. Because we are diverse, then the question can be diverse. So let's cherish our diversity by asking questions. Also, it's really important for us to understand the technique, how to answer the question properly. So considering the temporal answers, which are hypotheses, is really important. And it's not only one, but more than one should be cherished. And also proving method is really important if you wish to make the answer correct with this scientific way. And I think this is the essence of science advice. And I think we can call this as use-inspired basic science. So thank you very much. I wish to answer any questions afterwards.
Thanks a lot, Professor Cano, for this very good lecture and simple. Really, you raise a very good question. Why do we have diversity? Because it's the source of innovation, right? Okay, now we will start the discussions and questions answering and so on with Professor Cano for 30 minutes, half an hour, okay?
Sure, yes. With my tiring head, after my 20 minutes talk, it's not so easy to take every questions. So
you can start the discussions. You can start the discussions and some questions. Okay.
Yes, sure. So I was really liked the previous lectures, and I thought some questions already to all of you. So let me start from the one to Dr. Quraisha. So my question is starting from the what could be the balancing infection control versus economical activities in your country? So this is a very, I would say, deep question, I think, because the priorities are really different, but both we need as humans. So please turn on your mic, and if you can share your views. Thanks so much.
Yeah, that's if we had a whole day, it will take us a whole day. So I'll attempt to do a shorter response. And I think that many of our countries instituted some kind of stay-at-home policy very early in the response, and some of that was, you know, the complete unpredictability and about what to anticipate. And I think it was useful. It, you know, it was not just the stay at home, but restricting travel, large gatherings, and that was all, you know, part of the public health response and preparation for the increasing caseload. At the same time, when we look at some of the packages, stimulus packages and other things that we put into place, they modeled on macroeconomic policies. And I'm not an economist, so I can talk more about public health and less about the economy. But I think all of us who stayed, some of us were fortunate that we continued to receive our salaries. Some of us were frontline workers and at the coalface. And you see country after country, and as Maria spoke about, while we're Black and Asian minority groups bearing a disproportionate burden. So whichever country you went to, you found that the people who were first responders outside of healthcare, so other essential services being provided, were the ones with the least amount of safety networks and the least amount of protections. And these were the people who needed to use public transport. They are your day workers, they're your low-paid workers, and yet they were putting their lives on the line to ensure the rest of us continue to live our lives as normal. So I think that piece around how do you balance, firstly, we can't cocoon ourselves in the face of an epidemic or a pandemic. So how do you live and how do you live safely? And I think the piece that we've learned is, well, the virus is going to be with us for a long time. The vaccine was optimistically anticipated at the end of the year. It's not going to happen. So we got to use our non-pharmaceutical interventions that we have. And I think all of the speakers had lovely pictures of mask wearing and social distancing, etc. And we've got to get back to work. And it's that piece, I think, is how do we work? How do we get back to the new normal? But at the same time, balance, it's like as people, we have many things that make up our lives. It's not just preventing getting infected, but we also contribute to society in different ways. So when I think about the balancing livelihoods and lives, I'm thinking really about that as human beings, we are not as we were with the Spanish flu, but in the society we live in, there are many ways we can live safely. But at the same time, how do we also ensure that when we return to work, when we at work, those workspaces are safe and they mitigate against the spread of the virus and so on. And what I keep coming back to is the most vulnerable are the ones in settings, in workplaces that actually are overcrowded, where social distancing can't be done. But even within that context, we're learning about how do we make those places safer. As you say, in Japan, you have many challenges, including floods and tsunamis. And so that we can't stop life, but as we move forward, how do we make sure we're not cocooning ourselves on the one that's like we're just wasting oxygen in my mind if we do that. But on the other hand, how do we be active citizens? How do we have opportunities to contribute in whatever way? So let me stop there, and I'm happy to take on more questions or elaborate on that if necessary. Thank you.
Thank you very much for your kind answering. And I think now, yes, I will switch to Professor Remy. And in connection with what Professor Kwarashia answered, I think it's now great for us to elaborate about the connection in between differences, and which can be not only the interdisciplinary collaborations, as you explained, as the collaboration between microbiology and archaeology. Also, but it can be also some collaboration in between countries to overcome this pandemic and also preparing to the next pandemic. So would you please share some view about these potential collaborations or more needs of the collaborations?
Yes, thank you for the question. So yeah, definitely I give example from global alliances, whether for infectious diseases or monitoring. So I think it's a must. It's a vital. Sometimes people do the collaborations as I want to say as a type of being nice and having some inclusive and integrative policies, but now we are really in a crisis situation. So collaboration is, I want to say, it's crucial for everybody's survival. So as I give an example, we want to monitor emerging zoonotic risks from every country, especially those countries with high biodiversity, like Brazil, for example, with China, with so many climate zones and different types of habitats. They have deserts and forests and so on. So that there's so much richness there to monitor. So it's one of the vital things. The other one is, so again, we're talking in the context also of inequalities that Dr. Maria has brought. So at some point, we, like in a country like Egypt, which is a middle income country or middle low income country, we need collaboration to strengthen our research infrastructure, which sometimes may take a colonial aspect, is like you're needing support by a rich country. But at this point, collaboration is really on equal basis between all countries. So the richest country in the world still needs the most economically challenged country because of like one example is what Dr. Maria gave, is that you cannot produce a vaccine and test it on one ethnical group, or you have to test it on all different ages and races and so on. But also you want to see, like I give the example also with the sewage program, the global sewage program. So it's a global initiative, and similarly with the hospital one, it's not there yet. So the hospital project is not international yet. It's in different countries, but we are trying to make the same idea, is that every hospital in the world will report not just the patients. The patients is very well established in all countries. It's a public health, but in surfaces, like what is on the doors, what is on the walls, what is in the sewage, what is in the drinking water in different hospitals, because this is, it gives you an idea of what is going on and what is going to happen later. Again, I'm talking, maybe some people are out of the field. So we just take samples of water, samples of the wall, of the air, and see what are the microbes living there. And they can be very benign microbes. You can find in your apartment, for example, some bacteria that are carrying a resistance gene. So this doesn't sound like a problem now, but later these genes can be exchanged between non-pathogenic bacteria and others that are pathogenic. So this is the main purpose of all these programs, is to have a continuous prediction, not just at the animal level, but also at the microscopic level. So this type of collaboration, and lastly, but not, of course, not least at all, is collaboration by sharing information and data. As I gave the example, there is always companies that have know-how and private information. That's understandable, but in some situations, it's, as I said, it's not even ethical to start competing by hiding information and saying we will reach a vaccine or a treatment first. As you saw with COVID-19, the data are being shared mostly by third parties, by activists, but it's really important to have this shared on the governmental level, because, for example, we have things that we cannot understand, even like Germany, Spain, France, and Italy. They're very close to each other in everything, in economy, in population size, and they're all in Europe. But the dynamics of the epidemics have been very different. So we want to know how Germany managed to win, to overcome and to reduce the number of cases. So this kind of sharing information is very important. And I think it was successful in Europe at that stage, but it still needs to be expanded, because we're not there yet. And nobody thinks we are. Even I saw some questions about the second wave. The second wave is a term really specific to the flu, because flu is a seasonal virus. But for now, we cannot say we are beyond the first wave, we don't know if this is a kind of epidemic that comes in waves. It could be just, as Dr. Quraisha said, a new coronavirus that will be there forever.
Yeah, thank you very much for answering also to the audience question. I'm sure that the establishing some platform where we can share information would be very important to prepare for the next one too. So may I switch my question to Dr. Maria? And my question was, the utilization of scientific outputs are somehow, there are some discrepancy in between the people's expectation, where the scientific outputs are generalized one, but you may need individualized information as a general public. So how we can bridge them?
Yeah, that's a very interesting conundrum, isn't it? And I think something that in my practice has been really important, and I underappreciated during the first 10 years of my academic career, is the aspect of science communication. I believe that we were trained as scientists to work, to talk and to exchange information in this almost secret code that, you know, scientists all around the world would be able to crack the code, but it excludes the wider community. So I think that, you know, trying to find more effective ways of communicating complex, and I think Professor Kano, you did it quite well, and, you know, all the other presenters here, you know, these are very, very sophisticated systems, you know, that are very hard to be explained. And I think that all of you here were able to transmit knowledge to, you know, to a wider audience. I always try to put myself in the place of someone that is totally lay, that knows absolutely nothing. And I think that's the important thing. You know, the more we practice exchanging information in the simplest way, the easiest it is to get the population, because we are servants of the population. We are here to serve. So if you are here to serve, we need to be able to be understood by this audience. Otherwise, it's going to disenfranchise them and make our role more difficult.
Thank you very much for your very positive answer. And we may need to train ourselves more. Thank you so much. So if I may allow to pick up one question from audience related to that, the science communication, then the question is going to Professor Kwarashia again. The question is asking whether the scientific communication is successful in South Africa to get people understand about the science of the diseases.
Yeah. So I think there's two levels to it. The one is that, you know, in most of our countries, the initial response came from governments, and rightly so. Governments are there to protect their people. And I think there was a point at which governments needed to engage populations more. But in countries where there were extreme forms of lockdown, that transition didn't happen very easily. And so you started, you know, the fatigue comes in, like you can only stay at home, and you can't just stay at home, you need to get information. And so you started to see some of this dissent rising. And I think that's something I've seen in many countries. So in South Africa, there was, I think, you know, eventually we get to the point maybe about 2 to 3 months later where it now has to move out of government centralized control to more decentralized approaches. It's a point I was making earlier about we can't just breathe, we have to live. So how do we do that? And how do you take the fear that most of us faced a new challenge, a new threat, and the immediate human response is fear. But it is what